item that is blooming out there. So this hemialis likes to party, and he has some cyanobacteria inside. I think it's yeah, it's a cyanobacterium inside, cyanobacteria inside. So in A, we can see chlorophyll fluorescence inside of that same image. So you can see where is the chlorophyll located. And chlorophyll should be both coming from the diatom, but it should be also coming from the little cyanobacteria that lives inside of diatom. And this is very popular. Diatoms like to have cyanobacteria in them. But on a C and D, what you see is fluorescence that is specific to the pigments that are associated with that cyanobacteria. So this is phycocyanin fluorescence, this is phycoerythrin fluorescence. So it's pretty cool because just by looking at it in a microscope, you can shine different light, different excitation lights, and then you get emission at a specific part of the spectrum. So you can look at distribution of these cyanobacteria inside the hemialis. And very rarely, there's a paper, I didn't put in full, it's not a paper for you to read, but it's a pretty picture. So you can still hear when you give lectures about the fluorescence, because it's very rarely to find these really pretty images where you have 3 different types of pigments fluorescing in there. There's other organic components, they're fluoresce in a dissolved world. And we call them usually like sedum, everything, and a kitchen sink is in there. And that's a really bit complicated stuff, and I'll talk about that stuff. Oil fluoresces. And when that big oil spill happened in Gulf of Mexico, some time ago, I think that was during the grad school, so that was like almost last century, they didn't have tools to study it in situ. So they were grabbing our sedum fluorometers that you're going to be using today actually to follow that oil and oil spill as a function of vertical column. So that's kind of cool thing. And certain minerals in sediment also fluoresce. I'm not going to cover all these. I'm going to cover certain portions. So where does the fluorescence come from? This is the happy dude who discovered fluorescence. His name is Stokes. We've heard about him. And he said, okay, so fluorescence is a type of inelastic scattering. I don't think he called it that way, but he figured out that if he shoots a UV light on the rock, and this is a fluoride rock, that the light that is going to be shot back at him is going to have a different wavelength. So you can pretty much use this little simple formula, 2 formulas. Sorry, I said 1 formula is 2 formulas, the second formula. You can use this to calculate, what amount of energy shift is happening here, and see that as a shift in the wavelength, So once again, only certain wavelengths fluoresce. We have the absorption of that light on the higher energy, magic, and then the light gets out in a higher wavelength, lower energy. Some energy is being lost. And now the question is, what happens with that energy? This is just a reminder of what Meg went on Tuesday. You can have an absorption of the light that excites, those little electrons, and they can jump to different electronic. Is that a question?
Yeah.
Yeah, sorry.
No, it's okay. Is there ever a case where it's absorbed and then
Emitted in a different
emitted in
direction?
the opposite direction?
No, because you would have to have a source of energy coming from some other side,
There's a medium that was energized some.
If the medium was already energized, yes.
Right.
But the thing is, the problem is the fluorescence can happen only from the lowest excitation level. So in theory, it can be, so it can be already energized here. You can hit it, yeah. So if your electron, if your electron, I need a chemist. If your electron is already energized here, no? So you can, in theory, hit it with a photon that is this difference in energy. And it drops all the way to the basic state. So like not the lower vibrational state. In theory, you could get a shift to the left, but it has to be energized. It can't be energized to S1. I mean, you can have it energized to S1, hit it with the same photon, then you would lose some heat. No, that point it would be lower. So I think it just I think in theory, if it's not something else, if the energy is not coming from some other direction, you would make that happen only if it's in a little bit higher vibrational state. So then this energy would be lower than this drop.
But we won't realistically be seeing that.
You will, no. This is fluorescence. We go left. No,
Very
to the left. Remember that again. Okay, awesome. So one thing important to remember, and that's going to have to do something with photosynthesis and things like that. is that this fluorescence can always only happen with first excited state. So you can't excite something to S10 and then expect fluorescence to happen for that fall from S10 to S9, no? That usually, that release of the energy usually happens towards the heat. I don't know if ever happens to anything else but heat. That would probably like some kind of explosion or something, really, really super hot. The fluorescence, if it has to happen, it's always gonna happen in that the first step up, first excited state down, okay? How do we measure fluorescence? It was just a reminder so you guys remember connected to Meg. We can measure fluorescence in many different ways. Okay, so we can do fluorescent excitation spectrum, okay? So you do that by, you are looking at fluorescence at, what did I say here? Exactly 695. You just have a detector that is looking 695. And then you start exciting it from 400, measure, 405, measure, 410, measure, and you go to the full spectra. And for each light, you would measure the fluorescence. So you would get something that looks like this. Doesn't matter. Where here is your lambda of the shifted excitation wavelength. And this is your fluorescence at a specific wavelength where you were detecting. This makes sense? Okay.
Yeah.
So you have a detector that is sitting on one wavelength. And you're shooting blue, green, red, and as you're shifting the light, you're detecting, no? You have to kind of wait a little bit there.
And so what is the output or what are you detecting?
You are detecting fluorescence at a specific wavelength. You are detecting the transmission of light,
Which
transmission of energy.
Okay.
So for example, if I shoot, there was another question, is it connected?
yeah, but go ahead.
If I shoot, let's say if this is chlorophyll molecule. If I shoot light here, if I shoot light here, if I shoot light here, and if I shoot light here. And I keep on measuring at 695, the response, the transfer of energy. Is not gonna be always the same. I'm not gonna have a flat line. So this is lambda, this is my fluorescence at 685. If the transfer of energy is always the same, the molecule doesn't really care what kind of light you're shooting at it. You're always exciting it into the S1 level, you would have a flat line. Which means that the fluorescence is independent of the light source. Tell me.
So is the 695 the detector at 69.95?
Yeah, this is your detector,
Is that because
because I chose.
it has to fluoresce from the lowest energy state of S1?
No, I just chose, I just chose that.
Only detecting one wavelength.
I just detect one wavelength. I'm looking at the transfer of energy that is being absorbed across the whole spectra, and how does that energy being kind of getting dumped in the direction 685? And how does that energy getting dumped, and how much of that energy actually ends up flowing from that S1 to S0?
So wow.
Yeah,
I'm just,
Okay.
does everybody understand the mechanics of it? What am I doing here? Just mechanics. Don't know mechanics like an instrument.
Yeah.
Yes.
You're gonna go through and like start at 400, all the way down to 800, right? Yeah.
And you're always sensing that re-emission at one wavelength.
Just one wavelength.
And you could pick a different wavelength to
Of course.
sense if you
Yeah,
were
I
looking
could-
at a different re-emission spectrum.
Exactly.
But each sensor can only detect one wavelength at a time at the sensor.
My detector is really stupid. My detector can measure only one wavelength. Or I'm really, really interested what's happening at 685. I want to develop, I went to Mars. I went to Mars. I have sent only one probe to Mars. Only one probe, and the probe can detect light at 440. Okay? And then I have only so much, I have an emission that can go hyperspectral, but I don't have enough energy. So what I'm gonna do, I'm gonna go back and run an experiment to say, okay, I'm getting the highest response rate if I shoot it with 330 nanometers. I'm gonna save energy and just use 330 nanometers to detect it at 440. You're kind of checking how, this is like a stupid way of using it, but you're checking how does that energy getting transferred from the molecule all the way to that emission point. Let me go to the next step, it's gonna make more sense, okay? Fluorescence emission spectrum. This is a little bit upside down, okay? So now I'm gonna shoot one light source, one light, 440 meters, example. And now I'm gonna have a detector that's gonna be rotating through all the wavelengths. So that's gonna tell me where is that energy that I shot at gonna end up at. Okay, I'm searching where does that drop happen. Okay, so that's gonna give me that Stokes shift. Where is my Stokes shift happening? You can combine these two, and I'm gonna show you why would you use this for. It's really combined where you're analyzing one molecule. It's really good to combine it when you're measuring CDOM. And I'll show you how. When you have pile of things in there, you have no idea what they are. But you can develop these matrices that can help you differentiate different types of CDOM inside, because each of them is going to have a different emission excitation pair. Just bear with me now. A cheesy example of the fluorescent emission spectrum is pretty much if you would look at remote sensor reflectance, no? And the remote sensor reflectance, or in your your data from backscattering this morning. That's a cheesy thing of the fluorescent emission spectrum, because you can see the whole spectral response of the fluorescence, no? You're not shooting it only with one wavelength, you're shooting it with all the light. But in theory, if you would be shooting the light at 440 and detecting backscattering across the whole spectra, you would be able to see that the whole fluorescent peak spectrum. The bump that you saw in the end. Fluorescence is not easy. Like, just, we're gonna go slowly, tell me.
So with that first point, if you say you had like a pure sample of some pigment, something that fluoresces, that's
Yeah.
just super pure sample, if you don't excite it, if you excite it at too high of a wavelength, too short of a wavelength, you won't see anything. And if you excite it at too high of a wavelength, you won't see anything.
Yeah. Yeah, you're really, with doing the fluorescence emission spectrum, you're kind of exploring where that Stokes shift happens. And single excitation, oh, sorry, go ahead.
I was just wondering if we only can drop from S1 to something
S0,
low.
yeah.
Do you only get one fluorescence spike per molecule, or does it like if you're shining there you only have one transition basically? And
Mhm.
so in these spectra, can you see multiple spikes of fluorescence, or is there only one fluorescent signature
Per
per
molecule.
molecule?
Usually you would, I'm looking at this here, and this is a pile of different CDOM molecules where absorption is in red and fluorescence is in pink. You usually get one. I don't think the molecules, and this is my question, can fluoresce in two different spots. Like, I think like organic molecules that we would find. To my knowledge, usually you have a specific, I'm thinking phycoerythrin, I'm thinking like a bunch of these tryptophans and acids. Usually there's a specific, like a fingerprint thing. I don't think that, I think it would have to be a very complex molecule, like on protein level or something like that, which you would have in multiple fluorescence situations. Chlorophyll in certain situation, and I can cover that maybe later in discussion, might have two, but that's more because of the things that are happening within cells that are in the leaf, not in phytoplankton. Can we continue? And then I think some of the questions are going to be answered as we go. Single excitation emission, so you have, in theory, you excited one wavelength and you measure at a specific wavelength. And this is usually done after you do this recombination and excitation emission spectrum, so you kind of already know something about the properties of the molecule that you're hunting now. So for example, eco that you used yesterday, and eco that you're going to be using today, and eco that you're going to be using for the rest of your life, usually functions on the idea that we do know some chemical properties, chemical physical properties, fluorescence properties of a targeted molecule, in this case, chlorophyll. So we know that the highest fluorescence is going to be, you're going to get the really highest fluorescence. And we had those LED availables. If we shoot, for example, chlorophyll in 420 with the blue light and measure, detection somewhere in red. And we're going to be able to measure that fluorescence happening just with combination of two wavelengths now. If you're going to see today, you're going to be working with the CDOM fluorometer. CDOM fluorometer is going to be shooting light in UV and measuring detection somewhere in blue. If you would build a phycoerythrin fluorometer, where would your detector? So this is your phycocyan and this is your phycoerythrin. Where would your detector be? Look at the color. These colors are real. Would you put a green detector to detect this light? No. You build something that is orange. Here you would build something probably that is reddish or something. I don't, you really want to kind of find a way to do emission excitation spectrum of these molecules, and then you go and develop these targeted eco sensor, one wavelength. I'm calling the eco because it's the ones, one wavelength emission, one wavelength excitation to detect a specific molecule that you're looking for. And that's something that we're going to be using mostly in our field to study the ocean, like things in the ocean, targeted molecules in the ocean.
So then they will be target
Mhm.
and specific wavelength that is emitting and specific effect.
Yeah, you have the excitation wavelength, something that excites the molecule, gets it to either SX. You don't really care that when you excite the molecule, you get it from lowest stage to gazillion. You just care that if you shoot that light, you're gonna get ultimately whatever's gonna happen with that rest of the energy, you're gonna get this last transmission from S1 to S0 at the known wavelengths. So you have to know this and you have to know that. Yes.
Like, let's say, the fluorescence peak is static, or
Does it go?
changing in relation to the concentration of the component.
That's a fantastic question. I'm gonna answer it if we ever move past that third slide.
I guess I have a question about. We were talking about earlier, some things fluoresce and
Something
some
so.
things don't. Is it that some things don't, or that we can't? There's like a practical limit to measuring it. I guess I don't understand, because if everything that fluoresces is an inelastic scatter and the energy state changing, if the photon is interacting with something to a certain degree, is it not affecting the energy? So I guess I'm confused if everything's fluorescing and then we are only able to detect the signals that we have instruments capable of detecting.
So not everything fluoresces. And I'm just looking towards Meg, not everything fluoresces. You can have this drop going into the heat. You can have this drop going into the photosynthesis. So you can have chlorophyll that you know that fluoresces, it just doesn't fluoresce. And we're gonna get to that. So you can have molecules that you know that have fluorescence capabilities, but they're just not fluorescing for a reason. Because this drop can happen, But it doesn't have to happen. It can go into the heat, it can go into something else. It can be given to a friend that is sitting next to you, which is a different molecule. So not everything fluoresces, but the things that fluoresce, they don't have to fluoresce.
Things that fluoresce don't.
Yeah, I'm gonna get to that. It's so messed up.
It's
so messed up. Yes, Meg.
I just wanna talk that some molecules, when they're excited, have lots of options to vibrate. Yes. And release all that energy through vibration and transfer it as kinetic energy to the environment. Other molecules don't have so many options. They are like chlorophyll, like I'm shaped like this, that's it. Or maybe they're in a viscous solution or the solid phase or something like that.
So.
Yeah.
It's not, you're gonna see that not only depends which molecule you're looking at, even just with chlorophyll, let's stick with the chlorophyll. Chlorophyll might fluoresce, might not fluoresce. It will fluoresce differently if it's in different environment. If I put it in a methanol, if I put it in acetone, if I put it in water and so on and so on, it's gonna do different fluorescence. If it's in a cell, if it's outside of the cell, if the cell is happy, if the cell is sad, all these things will change and fluorescence is hard. And I'm gonna walk really slowly through all these concepts, why it's hard. And what is it vary, how does it vary for the chlorophyll specific? Is that enough?
Yeah.
And we can talk about fluorescence forever. Alright. We kind of went through this, and I'm gonna just jump really fast to CDOM fluorescence, so that's gonna answer some of these questions that we've had. So as I said, CDOM. We dump everything inside. It just says color dissolved organic matter. It's the same as we say live things. And now you're trying to, there's and the same thing as we say plants. Now all the plants are green. Can you differentiate a type of plant that is green? By just measuring that it's green, can you say which type of plant it is? No. This is even worse for CDOM. You have so many different components. They're dumped under that idea of a CDOM. It just tells it's a color dissolved organic material. And in this case, quite a lot of these things fluoresce, so you can use fluorescence to study it. And this was a paper where they did some analysis, and they just took available organic molecule that fluoresce and looked at the molar fluorescence, fluorescence that is divided, normalized by osmolality. And they looked at the absorption. So now this is normalized by the molar concentration, in here. And then they looked at the fluorescence in here. So you can see that shift happen. And it's kind of sitting kind of in a similar-ish area-ish, but not really, So I don't recall what all these things are, but I think there's caffeine inside. There's, I think, coumarin, and that's I think that's the thing that is in tonka beans, so it tastes a little bit like vanilla. So they were just trying to get all these different molecules to try to explain certain responses they're seeing in oceanic environment. But all these things are dumped in the same thing. We just call them CDOM. If you want to be fancy, then you would do that emission excitation spectra. Okay? So what is they doing here? They're combining two things. Okay, so let's go back to what I was saying. Excitation. So you have a light, and your detector is at 275. Excite. I mean, okay, so if you're measuring 275 and fluorescence cannot go right, but always goes to the left. So you excited 250 to 275, and you're looking, is there any fluorescence? Nothing, no? So then you go to the next one. You run the excitation, you measure this way, and nothing, oh, So we know when we hit whatever is in this water with the wavelengths, excitation, some light of 200 value of on 250 nanometers, we're gonna get some fluorescence at 450 nanometers. So you can do that and do these magical graphs. So this is called excitation emission matrix. This is from a paper by Zelinsky et al. And he took sample of water on the left graphs, 2 samples of water, obviously, and took them into the lab and measured this excitation emission matrix. And Meg was trying to do the same for the DRE last night, but she's going to do it again so you can have your excitation emission matrix for your CDOM, for your dissolved organic material water, so you can see how it looks like. But what is really cool, he developed a teeny tiny instrument that can do that in situ. You can deploy it on a little autonomous vehicle and stuff. And so what does that give you? What does that tell you? Well, it tells you so much more than just measuring it with that one wavelength, excitation of CDOM, what wavelength of emission of CDOM. You can start doing some analysis and start differentiating different sources of dissolved organic material, call it dissolved organic material in your environment. You can start, you can start separating based on the known values previously published, some terrestrial inputs, no? So this big peak here probably is coming with some kind of like riverine plume or something. This is really specific for some terrestrial humic acids. This is marine. This is marine humic acid, so probably this, what are we saying? It's going 307, 375, probably somewhere here we're gonna find marine, like marine humic type acids. These are protein-like, these are probably like zooplankton decomposing or phytoplankton oozing or something like that, no? So it's really cool, because now you can start looking at these emission and excitation spectra to say what CDOM is in the water and try to relate it to the processes in your ecosystem. And where do these sources of the CDOM is coming? Does this make more sense now in comparison what I was saying, excitation emission before? Everybody's nodding, Tom is still concerned.
No,
You're-
I'm thinking why would the terrestrial
Being
humic and fulvic have a higher wavelength at which it is emitting versus the marine? And then is there a linear combination of these where through the estuary you see?
Yeah, so I'm gonna overstate it. I'm gonna say I don't know a thing, but it probably can have something with the molecular mass and type of acid that's coming down in the water. With the marine probably, so terrestrial is usually fresher in a sense, marine is probably more photo-oxidized. So it's changing its chemical components, and therefore it's exciting and emitting. You have like chemical changes that are happening, and that's visible. These are really useful when you want to look at the photooxidation of your CDOM. Meg, am I going in a general good direction here? Ish. But you can, for example, use this to observe the processes, like for example, change of the CDOM due to the pH, change of the CDOM due to the photooxidation, or due to the different process, digestion of the bacteria, and so on. So why? I'm not sure. I'm not a CDOM person, that's why I'm not going too deep. But this is definitely a question that you can take to Meg. Ish. Am I saying anything terribly wrong? Okay, great. You're successful. Yeah, I mean, I just took the paper because I liked it because it has an autonomous version.
And just the second question, in those regions that are cut off, why are they cut off?
Okay, so where does the fluorescent go?
Where does what? Oh, shifting up.
Yeah, so it's cut off because if I'm shooting a light, so this is my excitation. If I'm shooting a light at 250 nanometers, you're seeing like, you're shooting a light and you can see emission on all these wavelengths. But if I'm shooting a light in 400, there shouldn't be any fluorescence at 275.
Also, if you shoot a light at like 250 and then you look at 250, you're just gonna see Rayleigh scatter.
Yeah,
And so you'll get the whole data set when we run these this afternoon. But you
Yeah,
see
and that's
Rayleigh
gonna have
scattering
Gaussian,
and the Raman scattering off water, which is huge and overwhelms everything. So a lot of times they just cut it off.
Yeah.
It's not look bad.
but it makes sense where there's nothing here.
Right, so there is a measurement there, but it's corrected because the theory.
Because yeah, I mean, if you shoot 400 at 250 and measuring at 250, then the energy transfer that you're looking for, the signal is gonna come, it's not gonna be coming because of this fluorescent Stokes shift type of energy transfer. It's gonna be because of the different processes, and that's not what you're looking for. These are not the droids you're looking for here.
Yeah.
Yes,
Which one? If that's the case, why is there a little slit in the right panel? Like, what does that get?
here,
Discontinuity.
this is probably due to the limitation of the instrument, so this is a real thing. Sample taken back to the shore. This is an instrument that they deployed in situ that probably has some technical limitations.
Okay.
Okay, and I can, the paper is in a Dropbox, in a shared folder, so you can check it out in details.
I might have missed this because I got
Yeah.
distracted, like reading paper. But what is the unit that you would have as the color bar?
Yeah, so the color, usually it's, the color bar is usually a Raman unit. And you can correct me, just leaving, but it's usually they use Raman units here for the emission excitation spectrum.
0.9 sulfate is the standard.
Okay.
So you would use a standard. So for all the CDOM fluorometry, we use quinine I know that's quinine sulfate in an acid solution. You can use also Sprite, but don't tell anyone, just get best about. Paper. But for CDOM fluorescence, you use quinine sulfate because it has a really well described excitation emission spectra.
Okay, and so then you just compare your emission spectra to that.
Yeah.
Show the intensity.
Okay.
Thank you.
They dropped the paper and it's kind of really cool. There's not too many people who are measuring the excitation emission spectra and actually try to use it to differentiate different types of organic compounds in the ocean. And it's really, really useful, but it's like, it takes a long time to do it. You have to know what you're doing. And there's really like, to do some kind of mass spectroscopy on the side to actually pair these things would be insane. You kind of like, taking a sample of water and do mass spectroscopy to determine different types of all these different types of organic molecules. It's really hard, take a long time. So this is kind of like a Cheeto way, like a proxy that you can use to say something about diversity of the organic material in your sample. We will be in this lecture room forever. So, I know, because it's a little bit different concept. You guys are not gonna be doing that. Meg is gonna be doing that. You're gonna have a single wavelength excitation emission spectra. Where you're gonna be looking at the CDOM fluorescence in your, riverine water, aka T, and you will be looking, how is that changing in your DRE sample and your two culture sample, well, actually, one type of culture sample under two different light conditions. You can then compare that to what Meg is gonna be measuring in excitation emission fluorescence spectra, and then we don't have that insight, but if she gives you this data, one good question would it be, now when you see the excitation emission spectra for your riverine water, are the choices of excitation and emission wavelengths that we have on this instrument the best one to use for the riverine estuary? Okay, so for example, if we go back, if I want to develop something to trace terrestrial acids in my sample, I would use probably excitation spectra, excitation wavelength to 250, and I would hit it somewhere here. I would measure the emission to 470 because it gives me the biggest, response. If I want to detect marine, I would hit it with 250 and then measure response to 400. And if I want to do protein light, I would have to just hit it with excitation light of 270 and measure the emission spectra at 360.
In theory, could you, if you have the same excitation wavelength, could you design an instrument that then senses at multiple wavelengths, so you can
Yes.
characterize it all at the same?
Yes, and they're eco sensors that do something like that. You just have to have a very narrow spectrum, very narrow detection window to not get the leakage, because you see how close these are. So yeah, so you could go maybe somewhere sitting around 270 and get a really, really narrow detection filters. I would put detection filter here, probably up high here, and somewhere really low here to separate these three substances.
And so you'd compromise the intensity of the excitation to for the instrument design, so that way you're using
Yeah.
only one excitation.
Yeah. That's exactly that. This is really like, if you want to go engineering, this is really useful. So yeah, so try to think about how you could optimize your CDOM. And I know it's kind of a little bit disappointing because we're just giving you eco puck, but you can actually use it to trace the water masses, no? So this is a really cool paper. I did something similar during my PhD, but this is has machine learning, so it's better. So this paper that just got published, and so they used, single excitation emission CDOM fluorometry, and they have it in micrograms per liter. Maybe they do some kind of crazy as conversion. Usually it's quinine sulfate in acid, and we use it as parts per billion. And versus salinity, they did some kind of chemical clustering to determine different water masses, no? And then when they plotted as a function of their glider profiles, over a period of time, you can actually separate really nicely with different types of water masses. So for example, if you need to separate your different samples and stuff like that, you can use these tools to, maybe it's not quantitative, maybe it's not great, but it can separate different water masses in your sample. And that's awesome. I think that's great. Because then it gives you, a step further down to differentiate different, ecosystem states, processes, so on. This could be upwell water, this could be river plume, and so on and so on. That's it for CETA. We're going to chlorophyll. Now it's getting hard. Did you use up really 1 hour? Now you should have already been at the, okay, doesn't matter. We'll do it fast. Chlorophyll. I have a chlorophyll molecule on my Christmas tree. I do. That's my Christmas tree, that's my chlorophyll molecule. It pays my bills and I like it a lot, and it's pretty awesome now. So this is a molecular structure of chlorophyll. So what you see on top, for all chlorophylls, and here is just showing kind of chlorophyll A and chlorophyll B, you have some kind of pyrolytic ring, and that's the thing that sits around the magnesium in the center. The differences between chlorophyll A and chlorophyll B are happening just there on the top. Chlorophyll C is a little bit different, it's a little bit less saturated, doesn't have a tail. But this tail that both chlorophyll A and chlorophyll B have is really that the phytol tail is really important because that's the thing that they kind of wedge inside of that lipid membrane structure. So when we were like hitting it with methanol, that's what we were releasing. We're moving away those lipids so that tail can come out and get flushed away. Okay? So these are not the only thing. You have, as chlorophyll starts getting to degrade, you have things that look very similar to chlorophyll. If I hit chlorophyll with some acid, it's going to start losing this magnesium. And we're going to call this pheophytin. If I hit it with some enzyme on top of that stuff, it's going to lose its magnesium, it's going to lose its tail, and it's going to become theophorbyte. And so on and so on and so on. If I don't hit it with acid, but I just hit it with enzyme, it's still going to have that pyrolytic ring, it's still going to have magnesium, but it's not going to have a tail, and it's going to be called chlorophyllite. All these things are processes that are happening inside of the cell, and chlorophyll does go through this conversion of different metabolic processes, and as it's dying, as it's going to sleep, as it's being eaten by things. For example, you can use chlorophyll fluorescence to look at the poop of a zooplankton, because chlorophyll gets eaten, and what is in your belly? Well, how do we digest foods? Acid, exactly. And the copepods also have acid. So what you can see is that you can analyze this copepod poop, and you're going to find lots of pheophytins, because that acid kicked out the magnesium out of the chlorophyll. So it's partially digested, and you can actually trace it. You can use that chlorophyll fluorescence because pheophytin will fluoresce still to detect kind of the fate of the zooplankton poop. There's so many cool things that you can do thanks to this magnificent molecule. So going back to that energy shift and stuff like that. So this is now plotted as a function of the absorption. So if you look at it, that's your chlorophyll absorption, no? There's two peaks in chlorophyll. You have the first peak of 440, and then the second peak that I always talk about, which is 675, which I like more. And they're actually indicative of two energetic jumps, no? Phytoplankton doesn't have anything to do. It's there. It's going to absorb that wavelength, that the energy that is coming to it in blue. No, it's going to excite it to S2. The difference between the S2 and S1, the difference in energy, and also different in these vibrational states, is always going to be released as a heat. However, when it comes to that S1 stage, that's when magic happens. The release from S1 to S0 can go to many different things, but if we're looking just as a molecule, just at a molecule or chlorophyll, it will be released as a fluorescence. And that's that shift here. So what is fluorescing is always this secondary peak. I mean, if the energy comes from the first peak, it happens here. So it's always happening here. So all these little lines are just energy jumps from different vibrational states up, heat still, but this jump is going to be happening as a fluorescence.
Do you have jumps from S2 to S0 as well?
You would if you would change the molecular structure of the chlorophyll, and there wouldn't be chlorophyll anymore. I mean, you have jumped from you can't jump directly from S2 to S like to the basic state. It has to go to S1 and then it has to go to S0.
Or it has to cascade?
It has to cascade. Okay. So, like if this jump in energy is just happening exactly from that basic energy level exactly to energy level S2, where we would see that as absorption would be just like one really super narrow absorption band, But because these jumps can happen on many different vibrational states, that's why we see this beautiful Gaussian, That's your Gaussian. And the same thing happens here in the fluorescence, shown with pink, that's your also Gaussian of your fluorescence. Because the release can happen in many different energy states of of the basic of S0.
So any measurement of chlorophyll is gonna require an excitation, or is gonna require light to enter it, right? So like, could you ever observe chlorophyll without exciting it? Like if you're just measuring absorption, right, you're still shooting light through it.
Yeah.
And so it's being excited.
Yeah.
Like, do we ever actually observe it and it's not excited phase? Or is it always excited?
It's always, the
That's
absorption is the
nature of light,
excitement.
Right, yeah, right.
Yeah,
okay.
absorption is the excitement. It's just the question was where that energy is gonna go. I'm talking about the molecule by itself. When we drop this energy to the first level and this chlorophyll is in the cell, there's so many different things. There's many choices that Meg is talking about. And I'm gonna talk about that coming up next.
Sweet.
Yes,
So we couldn't measure there'd be no way of measuring. You're saying the jump to S2 is then released as heat.
mhm.
There's no way we can't measure that in the infrared because of the heat absorption properties of water.
No, but there's a different way of measuring that. By using fluorescence. Again, I know, so much information, so little time, okay. Fluorescence of the chlorophyll measured in a specific wavelength is a function of excitation light at a specific wavelength, no? Absorption of the chlorophyll and this weird thing there, okay? So we go back here, makes sense, no? You have light that is hidden at a specific wavelength, you have absorption of your chlorophyll, your spectra, and then you have this weird thing there, and that weird thing there is called oh my god, it's
It's Friday.
quantum yield of the fluorescence. Okay, quantum yield fluorescence. Quantum yield of fluorescence is it's a factor that tells you how much of that light that is absorbed will be released back as a fluorescence. Okay, so we can resolve that further. We can start pulling things apart. The fluorescence of the chlorophyll measures specific wavelength is a function of the light at a specific wavelength, and then we can start tearing apart this absorption of the chlorophyll and say it's a function of chlorophyll specific absorption, which is your absorption spectra divided by chlorophyll times chlorophyll, which one, and times fluorescence yield. Okay, so we're now in solvent, okay? We have a molecule of chlorophyll. There's not a living soul close by. There's no membranes. There's nothing happening, okay? We put that light level that we know and shoot it at absorption. Let's shoot it at a molecule of chlorophyll. We know what is a chlorophyll specific absorption. We just know because that there's a chlorophyll molecule, one, and we know that absorption. We know this. This chlorophyll fluorescence yield of fluorescence is a very specific thing that people have measured. And depending on the solvent that chlorophyll is in, so remember, still talking about the molecule, that will change. So the amount of light that is absorbed that by the chlorophyll is going to be switching back at you as a fluorescence is going to be different in the different solvents. We use acetone usually, we dump it in acetone. It's 0.33 in acetone. And methanol is something else. And so you can use that now to measure your concentration of chlorophyll. So, because you can solve for so many things, no? So you can build an instrument, this is, this, 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 you have a measurement of this, and you can get your chlorophyll concentration. Because your absorption per molecule chlorophyll is static, it's well described. So turn around this formula and you make a chlorophyll fluorometer we're going to use for chlorophyll fluorometric measurements, because that's a very stable relationship, no? You maintain a constant A, you have a constant quantum yield because you know what the solvent is. There's always that the chlorophyll specific absorption doesn't change because you know how the chlorophyll looks like, how does it absorb, and you can derive chlorophyll molecules in solution. So you can have your instrument and you can have extracted chlorophyll, you get it online, spin it, you dissolve it in acetone, you make a serial dilution, and boom. You have a factor that allows you to measure fluorescence in one wavelength and calculate concentration of extracted chlorophyll on the other side. Fabulous, no? We're good. However, I said that there's other chlorophylls in there. I really don't care about the other chlorophyll. We kind of do, but we really want to know about chlorophyll A. And going back to that spectra that you were showing this morning of the backscattering, we're talking about some shoulders around it. And these shoulders might come thanks to the fluorescence of chlorophyll C's and chlorophyll B's. They spread that shoulder. And depending on the phytoplankton structure that you have in your sample, the presence of chlorophyll C and chlorophyll B will change. And that's not enough. In the same way that chlorophyll A degrades, chlorophyll B degrades, and chlorophyll C degrades. And all of them will still fluoresce. And they're really kind of not great. So what we're going to do, we're going to build an instrument that has an excitation somewhere here. And then we're going to use an instrument that has a really narrow band of detection and just measuring really, really narrowly the chlorophyll A fluorescence. We don't have that kind of instrument in the lab currently. So we're just going to go through it slowly. We're going to build an instrument that has a whatever excitation here. We're going to put a detection starting kind of like here. And what we're going to do, we're going to measure the fluorescence in our solution of chlorophyll, which is an acetone. Then I'm going to remember, oh, Ivona said that if I drop some acid, I'm going to kick out that magnesium out, okay? So I'm going to drop some acid in that solution. At that point, all chlorophyll is going to start kicking out the magnesium out. So everything that is in there, if I measure it, is not going to be a fail. Is everybody following? Okay, so I'm doing the second measurement. So all my fluorescence is now associated with fail or fail things now. I know the quantum yield of fluorescence for these guys, so I'm going to calculate the fluorescence, the concentration of phyllophytin, concentration of chlorophyll, and the difference in these two things is going to be chlorophyll. Because in the first solution, I had chlorophyll plus fail, degradation pigments of the chlorophyll. In the second solution, I killed all the chlorophyll and I made it phyllophytin. Okay, so one minus other is chlorophyll. Because I know how to correct for that quantum yield of fluorescence of each of these specific things. That's going to solve the issue that I have with phyllophytin if I have a very narrow band detector, but I don't. And it doesn't solve the problem of chlorophyll B and chlorophyll C. They're still going to be in there. And you have to know your sample, because if you have a sample with lots of chlorophyll B and chlorophyll C, they can mess up your chlorophyll fluorometric measurements. Most of the times, your chlorophyll A is very fluorometrically measured, is very comparable to our golden standard, which is HPLC. where we separate pigments individually spectra, we individually based on their peaks, we can, like measure each of them individually. What authors have done here, they have compared how does that photometrically chlorophyll measured chlorophyll compared to the HPLC measured chlorophyll. And they have found a relatively good comparison. Here you have from 0.2 to minus 0.2, good comparison. So this is the difference as a function of average. Does anybody know what's the name of this type of graph? So like we usually, when we compare two things, what do? How do we plot them? We have thing one and thing two now. And we want it to be linear. Yeah, this linear regression. This is called Bland-Altman plot. So what this Bland-Altman plot is doing is plotting thing one minus thing two as a function of average. And why are we plotting that stuff? Because if both of the things are the same, they're going to be on a straight line, regardless of the concentration that you're encountering. But if you see things going down here or down here, you can start talking about the biases as a function of concentration. So I can see that there's certain trends happening here. There's like this thing going up here and this thing going down here.
So in this one, when it's going up, that's saying that the H chlorophyll A is getting higher measurements than the F chlorophyll A.
Yeah, so it means that HPLC based chlorophyll, which is our golden standard, is seeing more chlorophyll than fluorometric chlorophyll. And this trend here in gray is saying that HPLC is seeing different, is seeing less chlorophyll than fluorometric chlorophyll. This is happening with this chlorophyll B in your sample that is polluting your fluorometric measurement. This is happening with this chlorophyll C. You have lots of diatoms. There's lots of chlorophyll C here. If we would measure HPLC chlorophyll, this would be probably the trend that you would see.
So this figure just tells you whether the fluorescence is overestimating or underestimating.
Yeah,
and
are
goes
you-
into your standard.
It's telling you, are you actually measuring only chlorophyll or are you picking up some other stuff? Because we know that we have a limitation in fluorometric measurements of the chlorophyll when they're extracted because of the presence of either phytophytins, degradatory pigments, or other chlorophylls. We can't make this detector window so narrow, we just cannot. So we have certain errors, and in certain situations, depending on the phytoplankton community structure and presence of different chlorophylls, you're going to see these trends where your fluorometrically derived chlorophyll is going to deviate from the truth. Yes.
I was surprised when you said HPLC
Mhm.
and the fluorometric methods match, when I'm coming from a legal context. So that line, as we go further.
Yeah,
makes it
so,
really
Like a-
right.
Yeah. No, what I wanted to show is this. This is the match. This is the fancy for the stats people, uncertainty people, this Z-score is where you want to live in, but. So gray stuff is good match. This is for the ocean environment. We find a really good match. In lake area, the paper is in a folder. Check it out. It's really good. We just published it now. Amy did an amazing job of comparing those two things. Why don't we always why don't we always use HPLC? Why are we using chlorophyll fluorometric? Why do we want to do that? You see where we are. We're nowhere. And it's lunchtime. Chlorophyll fluorometric, so extracted chlorophyll and acetone is fast, it's cheap. It's $1.50. HPLC is around $100, €100 for Europeans. I did that conversion for you. So we were Amy was trying to do this paper to see in which conditions can we just like start using chlorophyll, fluorometrically derived chlorophyll, so the one that we extracted with acetone for validation of the satellite, and we know that we cannot use it in lakes. And yeah, check out the papers in the Dropbox. Okay, but what I wanted to get to you is like, you're going to be doing this today. You're going to be extracting your chlorophylls. You're going to be dropping it in acetone. and you're gonna be looking at the measurement. What we're gonna do, I'm gonna talk to my colleagues now, we're gonna probably take a pause. And go now, this is a good place to take a pause, because I mean, we're gonna be here until midnight. And we're gonna go, all of you are gonna get a little bottle, and we're all gonna go to the dock, and everybody's gonna lie down on the disgustingly wet dock and sample in the same time. So what are we gonna do with that? We're gonna use all the same methods, very similar volumes and stuff. We're gonna look at the variability that it can be then probably, because you're gonna be looking at the uncertainties and propagating uncertainties. Looking at what is driving the difference, and you're all sampling the same time in the same spot. Is it natural variability, or is it you messing up along the way? Okay, so this is the first thing you're gonna do. And then we have to get to biology. So we're still that chlorophyll is still floating in acetone. We haven't even gotten to biology. I'm sorry, I'm like, yeah, I just like this is always a hard lecture.
So are we coming back in after?
We're gonna be going to sample, go have lunch, put our samples there, go have lunch, come back here and continue.
Okay, so I can leave my house.
Yeah.
That Yoda quote is so good.
Okay, so the first step of this lab today, everybody's doing everything the same. After this first collection, we're gonna leave these bottles in Mitchell lab. Everybody's gonna label the bottle. If you want, put the bottle in your pocket and click it. If you don't mix up, there's gonna be 19 bottles. Okay? We're gonna come back and you're gonna be splitting in 3 groups and do 3 things.
The handouts you just got are your 3 separate group handouts, so just read over your stuff.
Okay?
And do you want them to have it together?
No, they don't have it. So now we're just gonna collect water. I'm gonna try to go through the rest of this fast. It's gonna be a hard lab. There's lots of things to do. It would be great if tease that out to calculate the concentration of chlorophyll, because it's associated with absorption of the light, which is associated with chlorophyll. And what we talked about before, we have these things constrained. That's where our acetone is floating in a known solution, far away from any biology or anything like that. And this is relatively simple. Okay, we solve for certain things and we're good now. We just have to kind of think about what's the solution and light and stuff, and magic happens. And then there's the secondary pigments, but that's easy. That was easy. Now we're entering in a range of biology. And that's what's really bad. Okay, so chlorophyll doesn't float anywhere. It kind of sits embedded in those lipids. I said, like, there's that little tail that kind of wiggles it inside of those lipids in the membrane. It's actually part of these complexes. They're sitting in these thylakoid membranes. They're kind of these ear-shaped as little discs. There's a pile of little discs. They're called grana, and they're all sitting in a chloroplast. And that's happening if we're in a prokaryotic cell. So true cell, something similar to us, but then green. But if you're dealing with a cyanobacteria, they also have capability of photosynthesizing. They're not true cells, So instead of having all these organelles and everything, you just have, like, cellular membranes and things like that. And within these outside membranes, you're gonna find this accumulation, and also these protein pigment complexes that we call phycobilisome. And remember that I was told you, if we drop methanol on that, we extract lots of different proteins, but not the ones that are soluble in hot water. And this phycobilisome complexes are the ones they're really not soluble in water. And those phycobilisome type of pigments, phycocyanine, phycobilin, they're really hard to extract. And we can drop some hot water and then more hot water, pray to the gods of extraction, and we're maybe going to get them out. There's different methods, but it's important thing that you know that they're sitting in certain different types of membranes, and it's usually that little tail of a chlorophyll that is helping them sit there. And they're sitting inside of these protein complexes. So, if you look at this protein complex, I'm going to run really fast through this because this is not about explaining photosynthesis, just kind of reminding you that there's photosynthesis and stuff. So chlorophyll doesn't sit by itself. It's sitting with lots of little friends. They're called other pigments. And there's different types of chlorophylls, different chlorophylls that play different roles. And there's these antenna pigments. Let's talk about the antenna pigment that is chlorophyll, that is really there to capture the light. This is the chlorophyll A molecules. They absorb the blue light. They're called antenna chlorophyll. And sitting next to him are little antenna pigments, they're friends. Their friends are different pigments that exist in the cell. No, so depending on the phytoplankton type, you will have different friends. One cool stuff about all these little different friends is that they absorb longer wavelength, let's say towards the green range and red, but all these absorption peaks are kind of overlapping. And it's pretty awesome because once one absorbs the energy, it's going to toss it to another. And you're not going to have any energy loss. So it's going to be like, we have a really hot bowl of water. It's like, oh, you get it, you get it, you get it. And we're all like tossing gigantic amount of these hot water. And that's that energy that we're tossing around. It's pretty awesome, no? It's working really great. And so just because there's an overlap in absorption, and you saw that between the chlorophyll C, chlorophyll B, and chlorophyll A, it's just gonna tossing stuff, and now you can add some, I don't know, other pigments that they're tossing this happy energy, blah, 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 blah. What are they gonna do with it? Where is it all like going, It's going first into this photosystem 2. Photosystem 2 is inside of that, once again, membrane that we were destroying with our methanol, and that light that is absorbed is gonna go in here and is going to be used first portion, that step from S2. Now we absorb the blue light. We're going from S2 to S1. Heat, S1 to S0 is going to go towards breaking of the water. Okay? And then we're going to have a secondary absorption. There's lots of magical things happening. There's some electrons jumping around. Here we're going to get formation of NADPH. We're going to make some energy happen, and we're going to use that later during the night if we're plants to do the Krebs cycle and actually, take down that carbon dioxide. This is like the fastest photosynthesis that anybody has ever seen. However, if we don't shove that S1 to S0 towards the photosynthesis, we can dump it down as a chlorophyll fluorescence. Okay, so now our chlorophyll has absorbed light. It's not that the only option is now heat to get from S1 to S0 or fluorescence. Our third option is photosynthesis. It's very simplified, but that's what's happening. Okay? So, there's 3 fates of absorbed phytoplankton by that chlorophyll. First one is the energy that is used for photochemistry or photosynthesis. Okay? So let's go back to our S. So we're absorbed in blue. I'm drawing too much, but that's what I do. Okay, so we have absorbed, I think I should have ordered it differently. We have absorbed that light. We got that electron super excited, got to S2. I'm not doing any kind of vibrational stuff or anything. And then we're going to drop to S1. That's going to go as heat. That always goes as heat. Now we're here as S1. Because like in theory, phytoplankton doesn't need this energy. It's not good energy, but it's blue and it's there, and why not? You're in the ocean, there's lots of blue colors. So it's gonna take that, drop it as a heat. Now we're sitting at that S1. Once it get to S1, it can go to photosynthesis, photochemistry. It can go into vibration modes of molecule, heat, or it can go out as a fluorescence. There's three choices. This is again. This is now the chlorophyll is sitting inside of the cell, sitting inside of this great machinery. We have engaged that chlorophyll, so we don't have only 2 choices. Before we had choice fluorescence or heat. Now we have 3 choices. We have that beautiful photochemistry. If you look at average in the ocean, what happens with the fate of photon once it's absorbed by the chlorophyll in the cell of the phytoplankton on average? This is your distribution. And this is done by a really cool team using fluorescent measurement, but this insane fluorescent measurement that I'm going to touch upon a little bit. So on average, in a global ocean, 60% goes into the heat, 35% goes into photosynthesis, and 5% goes into the fluorescence. This is an average. It's super duper 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 highly variable, okay? Super highly variable. On everything. It varies with absolutely every single thing in the world. Okay. But there's only 100% of energy, no? So if I don't want to photosynthesize anymore, a percentage of energy has to go somewhere else. It has to go either to the heat or to the fluorescence. You can't shove it anywhere, like, under the table and put it away. It has to go somewhere. So if we can measure fluorescence, we can say something about these other relationships. So. Photosynthesis, fluorescence, forget about heat right now. Would anybody be brave enough to try to plot photosynthesis as a function of light? I think that one might be more easier, or fluorescence, but I think photosynthesis, I'm looking for somebody who I know that measures food. Okay, come, whoever, come, let's plot it here. Or you can tell me what to do, I like drawing.
I guess I was gonna say,
Oh, you wanna ask a question? I'm
yeah,
sorry.
by light, do you mean like, what do you mean by light? Is it
Like
radiant energy? Because I guess one of the things that will happen is as light gets too high, your photosynthesis will drop. Your
Okay, so,
fluorescence will raise.
so we are here on high, this
Yes,
is how we're starting this plot. This is low, no, this is great,
but
we know when we're high, so this is photosynthesis, activity. We get to high, it's gonna drop.
Right.
Okay, does anybody can fill up the rest of the story? You're doing great.
when it's low, it will be inefficient until it reaches kind of some optimal level.
Alright,
So
so-
it'll arc.
Yeah, it's gonna have kind of linear relationship and then it's gonna start kind of arcing, reaching a plateau when it's happy. And it's gonna start dropping down.
And then probably fluorescence won't really occur in that linear portion
Okay,
to a large degree. But once we've gotten to that high portion, we'll start fluorescing more.
let's go slowly.
Yeah.
Okay, so what happens in this, you have, let's imagine this, that linear. You have this kind of linear growth, you're curving, you're reaching some plateau and it's like, oh, and too much light, no, photosynthesis goes down. Walk me through this linear portion. For the fluorescence, and this is like anybody, we're guessing here, I'll show you the correct answer later,
How would it, oh, sorry,
go ahead.
I was gonna say, it goes down as compared to just like plateauing entirely, like if there's less photosynthesis then?
You go, when there's too much light, there's less photosynthesis.
Yeah, it just becomes.
So like, like, there's so much light, Like,
Oh, I'm sorry, yeah, it's like, yeah.
yeah, there's so much light.
Yeah.
You ate one burger, yummy, you ate second burger. The yumminess is not there, but that belly is getting filled, and I give you a third burger. No, exactly, so that's the thing, this is your typical, like, it's that you saturate it, so even if you increasing the input of the light. You will just be doing the same amount of photosynthesis because there's only so much industry in your cell, there's only so many mechanisms, there's like a limited number of photosystems 2, photosystems 1, transitional things and stuff, you can't. No?
So my guess would be that starting at the plateau.
Okay, we're starting from like that's great. So now we're starting from the middle. Okay, we're starting from a plateau.
No, at the beginning of the, yeah.
Okay, we're starting from the low light. What's happening with the fluorescence?
No fluorescence.
No fluorescence here. Okay.
No fluorescence, but as we will reach this plateau, fluorescence will start to happen because the energy needs to go somewhere.
Okay, so we're reaching the plateau. What happens now? Sorry, I didn't understand.
Fluorescence starts to increase.
Starts to increase.
Yeah.
Okay, we're gonna do this as a dash cell, dash. So fluorescence is increasing. And then it's increasing for the whole plateau, sorry.
Yeah. I would say, but there will be heat also with it.
And then what?
It's still light and then photosynthesis starts to saturate, it's when the actual signal in emission start to happen, fluorescence. So a bit like more like flat and then up.
And then up.
And maybe we went with fluorescence too early, so.
And I think fluorescence doesn't necessarily have to keep increasing
Yeah,
because a
that
lot of that will be
makes
released
sense.
as heat. It should go back down.
Yeah, it should go back down, but then the.
But not all the
starts
way to
emitting a bit later. So when. the
Okay, so Antonia, walk me through it. Well, let's go from low. Okay. It's gonna be, this one is gonna be wiggle. No, these are all good. Think about, we have 100% of the things. Heat. Heat's always gonna be present now. That's our biggest dump. So let's go. Okay, we're starting from low. We're starting from zero here. There's no light.
And then it's still zero,
Okay, so this is you.
still zero. Still zero. Still a bit more, and then when the curve is really like flat, then it starts
To go up.
to go up.
Okay, and then
And then.
It goes up?
It's a 5 point.
it goes up.
If when there's a then we go down. This
And
is too much,
Like here.
yeah. Because at some point it's.
Any other ideas?
I would think, I agree, but I think at that point, the fluorescence will go higher.
Or which point?
Yes, go higher.
Here is gonna go higher. Okay, so you are gonna be. I don't have different colors. Do I?
No.
Okay, you're gonna be straight line down here, so you're saying you're going straight. And then you go even higher,
Yeah.
and then you start dropping down, or you're going higher? Where do we go? Higher, yeah.
That's a lot of fluorescence. Love it. Dance party.
Alright. Any more? I'm running out of the. I'm gonna start doing little triangles now.
Line of just, it's, I know phytoplankton can undergo quenching, right, where like, if they're saturated with too much light, their fluorescence decreases. Would that be the point on the squiggly line that's past the photo inhibition part? Where would quenching be on this?
Okay, so this is your current productivity rate, and we were correct, okay? So you have the first portion where as you increase, forget about this, the dark respiration, we're not gonna go there, there's a freaking time. We are increasing the light, in the first portion of this thing, as you're giving burgers to the hungry cell, you have enough machinery to digest that light, no? To put it in everything, and this is probably the point where you're having that 60-35-5 ratio. You know, you have been yummy, awesome, there's, it's not like. Everything is saturated, but I can still keep it going, And then there's like so much excess light. So much excess light that you're getting to the point that you're gonna destroy everything what you have. You know, you're gonna start shutting up you're shutting up everything. You're just shutting up your prime productivity because there's so much light. And you start getting damaged. Your factory cannot just work anymore. And this is where you start getting this excess light, you start getting the photo inhibition of the prime productivity, okay? There's enough photochemical quenching. Where does it start? So this squiggly lines going down would be our non-photochemical quenching. So we have, who was the squiggly line? Antonia? She has found some, but it's not in the correct spot. This is your fluorescence. No, but it was correct. As you're increasing the light, you are increasing fluorescence as well. In that first portion, okay? So the increase of fluorescence is actually linear to the prime productivity. Which is pretty nice. Okay, we can measure fluorescence. Therefore, there's a certain portion of this light spectrum which plant lives where by the measuring fluorescence, we can say something about primary productivity, which is great because we don't have to do C4T. And we don't have to do oxygen stuff and all these different measurements are like. You know, this is direct measurement, okay? In one point, the fluorescence is gonna plateau as well. So thinking back into the cell and distribution of the photons here, where let's say we have 60% going to the heat, 35% going to the photochemistry, and then 5% is gonna go the chlorophyll. We're hitting here. Our photochemistry is staying stable regardless of the light, our fluorescence. In theory, it's continuing to increase, but you don't see that. We do parallel, it's gonna continue to increase a little tiny bit. The rest goes into the heat. And there's that point here where everything is too much. And this is your non-photochemical quenching. And non-photochemical quenching is actually indicative not only in the fact that you're turning off absorption of light, but you're also turning off your prime productivity. So if you can see non-photochemical quenching, which means that you do have chlorophyll, but you don't see fluorescence, that means that your cell is probably not photosynthesizing as much as it could. Your relationship between the light and time productivity doesn't hold it. And we can see that from fluorescence. Tell me,
Can I ask a quick question?
of course.
Is there a physical mechanism the cell does that makes it so that it won't?
Yeah, so remember those antennae? You know, the chlorophyll antennae who are responsible, we start shutting them down. You just like, no more. You know, they're turning off, they're just not absorbing it. I mean, they're not doing this. I'm sure there's like a physical chemistry process that do that, but in my head, they're doing this. They're closing their little umbrellas. And I'm gonna look and send you a link so you can actually understand the process better than you. In my head, they're closing their little umbrellas, because there are little umbrellas up there. So if we look, there's a portion of the time. So green is your fluorescence, red is my pathetic photosynthesis that I dashed last night in the middle of the night. You're gonna be parallelly increasing, your relationship is holding. You're getting a point where your photosynthesis is plateauing, but your chlorophyll is increasing just a teeny tiny bit. And now you're entering this quenching zone. And all these inflections and relationships and stuff can be measured in a different way using chlorophyll fluorescence. You blast a gigantic amount of light on them, and it gives you the FM, which is the maximum amount of fluorescence. You hit them with a little bit of light once they're just kind of like starting to do something, gives you that FO. The difference between these things is FM. And by looking at the relationship of these three things, you can say something about this thing, which is, once again, I forgot. What is the word that I said before? Oh my God, what is English doesn't listen to me today? Thank you, quantum yield. So efficiency, let's talk about quantum yield is efficiency of photosystem tube to do photosynthesis. This is a little bit more complicated than we're going to do in the lab. You have to have a specific instruments. This is called active fluorescence. I'm not going to go into that stuff, but in theory, you're still using the fluorescence. You blast them with the light. Usually when they're dark adapted, you blast them and you're going to get that maximum amount of fluorescence. Maximum amount of fluorescence. And it's just teeny tiny bit. And you take these numbers and you can get really, really nice ideas what's happening in the cell regarding photosynthesis just by using fluorescence. You have a question.
So where along that graph does the eco or sensor measure?
So
fluorescence?
where do you think it doesn't measure?
I guess it depends
Is-
on how strong the light is and how long it's been on.
And also depends what is the light history of the cell.
Yes.
So today you're gonna have two groups are gonna measure culture. One is measuring low light culture, just normal light, the left and the mixed lab. The second one was here in front of the blasting grow cells since last night. So I'm hoping they're gonna be here. The other one should be somewhere here. So we're gonna see, because we're always shooting the same light. In theory, your eco should be working somewhere in this area. But it also depends on the light history of the cell. Because if it has been like so freaked out, I'm gonna get you, it's gonna be so freaked out, you can blast whatever light, it's just like it's done. It's done, it's quenching, it's closed up, this umbrella doesn't want to talk to you anymore. So even if it has chlorophyll, you're not gonna measure any fluorescence. Yeah, go.
Can UV does degrade pigments? Right?
Yes, it
Yeah,
does, but that's a completely different process. We're not gonna go there. We're not going, no, oh God. Can I go through couple, can we do, no, can we write, I just wanna get the flow going. I'm gonna go to the next slide so you can see what I wanna talk about. No, is it something that you don't think that I'm gonna answer? Oh, like, okay, let's do it fast and then write down your questions so we can talk about it.
No.
Okay.
I just wanna know, like, what the heat releases really go on, like the. You have like your fluorescence, when you have your photosynthesis,
Mhm.
is the heat like actually doing something much the water? The heat range, it really, it's a really small amount of heat being released, you said like 60% of the
Mhm.
energy released. is
Yeah.
heat. So is there any like research or?
So in theory, so Meg said, Meg was talking about that heat release, it releases like as an infrared. And I don't know if anybody does measurement of the heat
Oh.
release of the fluorescence, I really have no idea.
Not very. They think about light going in the water and heating it up, right, and very detailed, and this is just like some pilot plan can get in the way of that sometimes.
Yeah.
Right, and the heat goes slightly through the top of the top.
Yeah, so you know how you can figure out this? If it ever heats up here. If it ever heats up here. When I lived in Maine, the only place where I was actually swimming is in lakes that are very, very dark. Because they absorb blue light. And that light, portion of that light gets released as the heat. And that's why it's warm. So you can actually see that visible, but I don't know if there actually anybody really measures that heat dissipation as a function of photosynthesis and stuff. It's much easier to measure all these other things, and then you can do the calculation of the amount of energy that is then dissipated. Probably does, but I don't know. I don't know, but I'll look it up for you and give you, okay, so just before you ask a question, there are if anybody else knows, please answer, because I don't know everything. There's these two things, these two papers are in your, so all the papers that I'm talking about are in your shared folder. This is Gorbono and Falkowski go into this in details, and probably in there, if there is a reference to somebody measuring the heat dissipation in some weird way, it's in there, okay? We're not gonna go into this. Am I helping or not? Am I saying stuff wrong? Okay. Let's go, so we actually get stuff done. Okay, so now when I told you all these crazy things that can happen. No, so it's the same formula, it's still the same thing. If I shoot a light, shoot a known light, add the chlorophyll. Like, chlorophyll's gonna absorb it. Depending on the. Thank you, I know it now, but everybody has to know. We can calculate chlorophyll, yeah? That was when it was sitting in the acetone, okay. Now we know it gets quenched, so here, it's a chlorophyll fluorescence in vivo. EcoPuck. Divided by the chlorophyll that I measured in acetone. During the cruise, some kind of cruising, never mind. And as you're increasing the light, and here indicated, measured as a far, this bright light. As we get, everybody's doing fine. So the chlorophyll fluorescence in vivo with the instrument is pretty much similar to the one that I measured extracted in chlorophyll until we reach certain stage, and then this happened. How do we call this?
No.
Non-photochemical quenching, okay? This is your indication. Okay, so we could use this thing, but it's varying here, okay? So the proportion of the light that I'm absorbing as a cell, my distribution of the light, I'm throwing less towards the fluorescence. So I can't really easily solve for the rest, even if I have this, because this is too variable in the ocean. Okay. We saw what for one molecule of chlorophyll know exactly the absorption per one molecule. If we have 3 molecules of chlorophyll, we know how that absorption is going to grow if it's in soluble form. But now we have it in the cell, we have it in this thing where it has lots of friends, there are other pigments, and we're shoving this thing around, and then there's packaging, there's all these crazy things that happen in the cell. So not anymore, we can do standardized. So if we take the A5 and divide it by here in this case 412 and plot it versus chlorophyll, we don't have that straight line that we would expect as we do when we have that chlorophyll inside of the acetone, because there's other pigments that are messing up the thing. So this relationship doesn't really hold anymore, no? And then, because of those other pigments and that thing that I was talking about, and then the light sensitivity and then nutrients and all these other things, when you compare your calibration from the factory, which is pretty much how much chlorophyll per chlorophyll fluorescence that you measure from your in situ instrument, and you do that calibration in lab with many different cultures, you're getting these insane relationships. They go from, I think this is like 0.5 to 6. It's so different because all these different cells have different pigments. They're helping chlorophyll transfer their light and modifying the quantum yield of fluorescence. They're also modifying this chlorophyll specific absorption. So this thing is so variable, like crazy, like crazy. So it's really hard for us to have this formula and use chlorophyll fluorescence to, measure chlorophyll as we did in soluble form, when we kicked out the chlorophyll outside, it's so variable. It varies on species. It varies the history of light and the amount of light. It varies if the species are happy or not. If it's stressed because of the nutrients, it will in fact impact that chlorophyll yield. It will, if it's cold, if it's hot, if it like, there's so many different things. They're super, super variable. So our chlorophyll fluorescence measured by that ecopuck is not anymore really relatable to the furthest extent to chlorophyll. And that's what you gotta do. So you will be given two cultures. One group is gonna be doing DRE. One group is doing culture of the Hitoceras, and third group is doing culture of Hitoceras. One culture of Hitoceras is sitting on a high light, second one is sitting on a normal light, and the area has everything inside. You know, like all the species known to humankind. So there's so many different variability. You're gonna try to do a calibration curve. We usually do calibration curve with tons of different things, but you guys have to sleep and want to finish up and have drinks and stuff. So you're gonna do just like 3 different dilutions, actually 2 different dilutions. You're gonna have a full sample, 2 dilutions, you filtered seawater. You're gonna extract chlorophyll in vitro, so extract it with acetone, measure, and then you're gonna parallelly collect ecopuck, eco VSF measurements of chlorophyll and try to make the calibration curve. As you're doing so, you're gonna also compare curves and tell us everything about it on Monday. And think about what are the implications of a different light conditions we put them in, or culture versus the stuff that is outside in a DRE. We saw that there's lots of diatoms, but there was definitely some dinoflagellates and other things that might be contributing to this relationship. Okay? Any questions now? I'm gonna have 2 more slides and then we go filter. Wow. Okay. More fluorescence. We can see fluorescence from space, and that is awesome. Okay. So sun shines the light on the chlorophyll. So we don't walking around with ecopucks or lightsabers and stuff. The sun is shining for us. That's how we see remote sensing, thank God, no? But if the sun's shining on this stuff, it's absorbing the light, it's doing photosynthesis, and once again, around an average 5% of that light should be going towards the fluorescence, and we should be able to see it from the sky. And this is called sun-induced fluorescence, no? So light source is sun, the chlorophyll is absorbing stuff, and it pops it out exactly what we would expect at around 683, 684, because that tends to, to the left from the second absorption peak of the chlorophyll. So a long time ago, people have measured that stuff and said, that's awesome, let's launch a satellite that has some bands. So the blue line here is indicating that chlorophyll peak, how it would look like modeled in water leaving radiance or remote sensor reflectance in ocean color, no? And this is your wavelength. So we're sitting there in that deep red, no? So this is how it would look like modeled. And so they said, okay, so what we're gonna do, we're gonna put a band here, a band here, and a band here. We're gonna fit a line from here to here, and then calculate this height. And then you're gonna call that fluorescence line height. And then this has been done so far in MODIS with Aqua and Terra, which are 2 different satellites launched by NASA 25 years ago. Not doing well. Maris, which was a European satellite launch, died. And then Olchi, both of the Olchis can have that capabilities, the Sentinel series. And before you ask, let me finish up the sense I beg you. And then you can start observing these things, this fluorescence from the sky, and you can start making maps like this. And it's really cool. So you have the one number can tell you a lot. It can tell you, once again, think about what do we say what fluorescence means. It means how much chlorophyll is there, so you can correct it for chlorophyll. But then the residual variability can tell a little bit about their light history. How much yum-yums do they have? Maybe even something about phytoplankton structure. You can even use it to calculate the quantum yield of fluorescence and start playing with some prime productivity estimates.
Yes,
I have 1 slide. Do you want to ask now?
I would, quick one, I
Yeah.
just wanna know why the band is shifted instead of being directly at the peak.
Exactly.
Why is it
Same.
Okay.
You know,
Did they
when Jeremy comes here, he was actually alive during the time. Sorry, joking. He was around, I think, when they launched Notice, and you can ask him why they didn't put this band on the peak of the stuff.
Do other sensors do it?
No, it's very similar to this one. It could be just something with the availability of the detectors or something. You have to always remember that, that we're looking at the ocean from way up there. And 90% of our signal is coming from the atmosphere. And I think, Patrick on Monday was talking about absorption and radiation and all these things. And I don't know if you remember that those oxygen absorption lines and those ozone lines and stuff. So this is in phase zero. This is what we get from phase. So your dots are your modus. And this is what we get from phase. So let me take it to, to this fluorescence spectra, and I'll explain you what this is. These are those three lines, there's third dot, sorry, three dots missing. And this is what phase is seeing this whole peak. So my problem currently is that there is an oxygen band absorption here that is destroying me, and we're working, getting better. I think they should be reprocessing as I'm talking to you, and it's gonna be better. But there's, this is a very sensitive region, so it might be that there's an absorption peak in atmosphere that I don't remember. And that's the reason why they moved that line, that measurement line from here to here. I'm a biologist. But what I wanted to show you is how awesome PACE is. So the dots are modus. The dots are modus. Yeah, you guys are laughing. I went to NASA so you guys can do this. I went to NASA 10 years ago. No. It's just like I did because I wanted to play with this data because I was done with dots. No, I wanted to play with the continuous stuff. So this is amazing, no? So for different spectrums, radiometry, this is like remote sensor reflectors, and look at this peak. Look how pretty it is. And now it's really stupid because we're still calculating this fluorescent line height. There's just like one dot. But look at this shape. What do you think that shape can tell you?
Could it potentially help with the different fluorescing chlorophyll pigments?
Maybe,
So
maybe.
they'll have to peaks.
Yeah, so what would you get if this, let's say peak starts moving to the left or to the right? And remember, like, we're just seeing the surface of the ocean. And this is signal that travels. So it can tell you how deep is in the water column, that chlorophyll layer. It tells you so many magical things. It can even tell you some other things about the pigments. We don't know. You guys have to figure it out. It's so awesome. Anyways, we can see it from space. We can do magical things. And that's it. I'm done with the fluorescence. And so one group activity, you have done it already. You have to filter the stuff. Split in 3 groups. Each of you have a culture, babysit the culture, do exactly as you're told. You're gonna go and make a serial dilution, and then the little dance starts. And we're gonna make it happen, okay? When we put the filter on that filter rack, we go filter, okay? Discussion about a group activity after this. Traditionally, at the end of the first week, after dinner, we bring some oysters, we bring some cheese, wine, hide away from the interns and just have a lovely night. It's disgusting out there. We can do it tonight, and probably it's gonna stop raining, but it's still gonna be cold, and I don't know if we can start a fire. But oysters have been picked from the aquaculture facility that is owned by Stuff and donated to the class. We have 60 oysters. I'm looking at, or we can transfer everything to Sunday and risk losing the oysters to harm vagabonds. We have 6 oysters. We have
So they're all Sunday.
Huh?
So.
Yeah.
Actually, it sounded like,
Sunday.
speak out, if you guys disagree, but something. Pretty preferred.
Well,
Sunday and Friday.
we were talking about doing something on Sunday. That might not possibly be-
No.
We don't know.
This would be like a late evening. Alright, that's the game. Yeah, if you guys are going to Acadia, if any groups are going to Acadia, Sunday's gonna be hard. This is where democracy has to stop. Somebody has. I should not have given you that.
I mean, if we have the oysters, if we know we're good today, we could just do it today.
Yeah.
Let's
I
just
mean,
do it
don't
today.
be pushed it into eating oyster,
I can't eat fish, so I
Yeah, no, we're gonna have some cheese and grapes, and if somebody goes in the store and buys it. And wine and stuff, it's just like a little thing. No.
Yeah.
Yeah.
We wanna just celebrate tonight.
Yeah. You have some amazing stuff. Okay, go. Please go, Ollie, let's get it done. Thank you. Ask Millie questions later. I mean, I have more slides.